Dear student, Assalamu alaikum. I hope you people will doing well. Today is our first online class. I will try to explain each and everything in a very easy way. However, if you have any query or problem related to this lecture, you may directly ask in our created WhatsApp group or you can email me. I will try to respond you on time. If you remember, last time we have been discussed about taxonomy and its importance in botany. Further, we explain different system of classifications. If you remember, we explained four different system of classification that were artificial system of classification, which were based on arbitrary or at least easily observed characteristics that were habit, color, number of similar features or characteristics. The expert of this system was called herbalist. Then again we explain natural system of classifications. This system of classification was based on morphology and reproductive characteristics. The expert of this system was called naturalist. Another system of classification which has been introduced in taxonomy that was phylogenetic system of classifications. This system of classification was based on evolutionary, particularly Darwin theory of evolutions. Then another system of classification which is also called modern system of classification has been introduced. We explained that this system of classification is latest system of classification. However, this system of classification is still under change depending upon the new available information. Modern technology like electron microscope and biochemistry are employed to establish the relationship between or among plants. This was just overview our previous lecture about our previous lecture. Our today topic is taxonomic evidence. It's mean that what are the main evidence on which we can easily classify the taxon are helpful for classification of plants. The taxonomic evidence may include different parts like root, stem, leaf, internal parts, and biochemical characteristic. Remember that based on taxonomic evidence, foundation of modern technology become possible. The content of our today lecture is introduction. It means that what is taxonomic evidence, morphology in relation to taxonomy, anatomy in relation to taxonomy, phylogeny in relation to taxonomy, embryology in relation to taxonomy, cytology in relation to taxonomy, phytochemistry or phytochemical in relation to taxonomy, ultrastructure in relation to taxonomy, genome analysis and nucleic acid hybridization in relation to taxonomy. These are the main features on which foundation of modern technology become possible. Now, we will explain these features one by one. Taxonomic evidence or taxonomic characteristic are the taxonomic attributes that can be used to provide the evidence from which relationship the phylogeny between taxa are inbred. It's mean that what are the main characteristics which show resemblance within or between the species. You know that for a system of classifications, a particular or specific rules are followed. For example, Species showing similar characteristics are placed in a single genus. Similarly, a number of genus showing similar characteristics or features are kept in a single group. Similarly, a number of groups are kept in classes and a number of classes are kept in a single file. These classifications are actually based on taxonomic attributes. Okay, morphology in relation to taxonomy. Before going to taxonomic evidence, we must know about plant morphology. Basically, plant morphology is the study of outer appearance characteristic of a plant, or we can easily say that the study of external characteristic of a plant species is called plant morphology. It mainly deals with the external characteristic of a plant, such as habit. What are the habits of plants? Either the plant are an annual or biennial, or the plant are living in moist site, or the plant are living in dry site. Similarly, the plant are living in land, which is called terrestrial, or the plant are living in water, which is called the aquatic plant. Similarly, root characteristic or root structure, either the root are monocot or dicot. Stem characteristic or stem structure, which may be hardwood, softwood. Stem habits, we can say the herbaceous plant, we can say that the plant are tree, or we can say higher plants. Bud structure, leaf structure, and either in leaf the venation is parallel, reticulate, inflorescence types, flower type, androecian type, stamen types, carpal structures, wheel types, fruit types. Like in fruit types, maybe the fruits is bilateral symmetry or asymmetry. Similarly, the different seed types. So these are the morphological characteristic which provide the basic language for plant characterizations, classification, and identifications. Remember that morphological data is very crucial for taxonomic studies. Anatomy in relation to taxonomy. Plant anatomy is basically the study of internal structure of a plant. It is often called phytotomy. The anatomical evidence which are useful for plant classification are included as wood cell type, size and shape, cell wall pattern, stellar pattern, vascular bundle type, epidermal type, trichome type, and venation type, etc. These anatomical evidence provide the basic platform for plant identification and classifications. The application of anatomical data to phylogenetic problem is of great value. Therefore, anatomy poses a significant role in plant taxonomy. This slide is about palynology in relation to taxonomy. So, what is palynology? Basically, palynology is the study of pollen grain or spores. Pollen grain come in a wide variety of shape, size, and surface, marking characteristic of the species. Most, but certainly not all species have a spherical pollen grain, but there are some certain species which have around pollen grain. Basically, pollen grain is the carrier of male gametes. Pollen grain is one of the most important characteristics of plant species which help us in characterizations and identification of various plants. Different plant species have a different pollen grain type and can easily be classified on the basis of pollen unit type, pollen grain shape, pollen grain symmetry, pollen grain nuclear state, exon stratifications, exon structure, and aperture type. In addition, different species have different pollen grain with different nuclei. For example, in Magnoliidae, the pollen is nucleates. In Caryophyllaceae, the pollen is trinucleate, and in Ericaceae, the pollen is in a tetrad form, and in Asclepiadaceae, the pollen remain is pollinaria. So, going to next slide, we must know about what is aperture. Basically, aperture are the place where the pollen tubes is able to break through the pollen wall. This is basically the softest and thinnest area of pollen grain from where the pollen tubes is able to break through this particular wall. Now, what is pollinia? Basically, pollinia are a plural word. Its singular is pollinium. A pollinium is a coherent mass of pollen grain in a plant that are the product of only one anther but are transferred during pollination as a single unit. This pollinia can easily be seen in a plant such as orchid and many other plants of milkweed, particularly family Asclepiadaceae. The coherent mass is basically the consistent mass of pollen grain which is based on a single unit. Okay, G. This slide is about embryology in relation to taxonomy. Embryology is the study of the successive stage of sporogenesis, gametogenesis, and the growth and development of embryo. The sporogenesis is actually the formation of spores by a plant, where the gametogenesis is the process in which cell undergoes meiosis to form gametes. Similarly, embryo is the part of a seed or bud that contain the earliest form of a plant, root, stem, and leaf. It means that the earliest form of plant is called embryo. The basic evidence which play a significant role in a taxonomy are included as anther, loculus or loci, which is the layer of cell lying beneath the epidermis of the wall of the anther, arrangement of anther wall formations and endothecium type, embryo sac development type, embryo and embryogenesis type, endosperm type, integument number, and structure type, etc. So these are the main features which play a crucial role in plant taxonomy. This slide is about cytology and its relation to taxonomy. So, plant cytology is the study of internal organelles of a cells, or we can easily say that the study of morphology and physiology of a cell is called plant cytology. To determine the origins of groups and to understand the evolutionary history of related taxa, plant cytology possesses a significant role. Some evidential characteristics of plant cytology are chromosomal number, chromosomal meiotic behavior, polarity levels and types, a chromosomal characteristic, etc. So these are the basic cytological features or characteristics which play a vital role in plant taxonomy. Now, phytochemistry in relation to taxonomy. As I have already explained that phytochemistry or phytochemical is the study of different chemical which can be obtained from different plants. So the chemical study of different plants are called phytochemistry. The different chemical which can be obtained from different plants are flavonoids, terpenoids, carotenoids, polysaccharide, alkaloid, amino acid, fatty acid, aromatic compound, C3 and C4 photosynthesis compounds. So these are the basic chemical which can easily be extracted or obtained from different plants. Remember that majority of plants and crop plants are C3 plants, referring to the fact that the first carbon compound produced during photosynthesis contain 3 carbon atoms. So here we must explain that what is the C3 and C4 photosynthesis. Please keep in mind that in C3 photosynthesis, first we have to talk about C3 photosynthesis. C3 photosynthesis occur in mesophyll cells. First, C3 photosynthesis occur in mesophyll cell. The second characteristic of C3 photosynthesis is C3 photosynthetic plants are confined in cool and wet area. Remember that 95% of green plants are C3 plants. The rate of photorespiration in C3 plants are also higher. Now, what about C4 photosynthesis or C4 photosynthetic plants? So, C4 photosynthetic or photosynthesis occur in mesophyll plus bundle sheet cells. These plants can be found in dry regions. 95% of the green plants are C4 plants. Photorespiration is completely absent in C4 plants. Remember that the photorespiration in C3 plants are very high, while in C4 plants the photorespiration is absent. Now, ultrastructure in relation to taxonomy. Remember that with the invention of electron microscope, a dramatic change occurred in taxonomy. Because we can easily study the internal part or external part of a plant which cannot be seen through naked eye. For example, sometimes there are a plant which show very clear resemblance with each other. We cannot differentiate through morphological study or other relevant approach. In such cases, electron microscopic study play a significant role for plant identification and classification. So it means that electron microscopic study play an important role in plant taxonomy. For example, Behenke et al. Study more than 1,500 species belonging to 280 family. They found 3 different plastic in these species. Similarly, Favela and Girola reported dilated cisternae in phloem parenchyma of foliar veins in Brassica chinesis. These dilated cisternae were also found in the Caparis species, which was invented by George Jensen in 1977. So the microscopic study become possible through electron microscope invention, and with the electron microscopic study we can easily classify 10 plants which show a very close resemblance with each other. Now, genome analysis and nucleic acid hybridization in relation to taxonomy. Genome analysis and nucleic acid hybridization also play a significant role in plant taxonomy. Genome analysis and nucleic acid hybridization contain identification, measurements, arrangement of genomic features. Genomic feature is basically referred to define segments of genomes which often code for protein synthesis and RNA. The common genomic features are included as genes, ribosomal RNA, and transfer RNA, etc. Genomic analysis has a very crucial role in understanding genomic relationship among species. These were all about taxonomic evidence and its importance in plant taxonomy. Now for you people, there is a question from our today lecture. This question will consider as a quiz question. You have to find the answer of this question and can send to me on my email ID. The question is, why taxonomic evidence is important for modern taxonomy? Describe in its own word. I am repeating again. Why taxonomic evidence is important for modern taxonomy? Describe in its own word. This lecture material was obtained from 3 different books that are Taxonomy of Vascular Plant, written by Leverance, An Easy Approach to Plant Systematic Anatomy and Development, written by Zaimgi, An Easy Approach to Plant Systematic Anatomy and Development, written by Akhtar Hussain Shah. Some material for today's lecture were also obtained from internet. If you have any query or question, you can ask me through my email ID, that is adam.khan345@yahoo.com, or you can ask me through our WhatsApp created group. Take a good care.